Okay, I think I'll just get started. My name is Russell Hanson, and it's a pleasure to be here at OSCon Data. I'm going to be talking about synthetic biology and data-driven synthetic biology for personalized medicine and clean energy. And this talk is going to be a little bit broad because it's difficult for me to assume what the background of the audience is. Who here knows what synthetic biology is, or has? Okay, a lot of people have heard of what's it. Who wants to learn more about what synthetic biology is and doesn't know what it is already? Okay. Also a lot of people. So synthetic biology is basically using different synthetic constructs in biology and assembling them into new things. So kind of like in electronics, you can take an AND gate or an OR gate or a transistor or an integrated circuit and make it into something new. In synthetic biology, you can take different little biobricks, as they're called, and make new bacteria or viruses that perform a particular function. So this is a very new field. In some sense, it may have started around 2003, 2004 with the iGen competition, which is the competition for genetically engineered microorganisms. So my interest in this area is particularly as it relates to potentially cures and treatments for diseases in a personalized medicine setting. So it's becoming cheaper and cheaper to sequence people's genomes and figure out what exactly is wrong with them. So for instance, in breast cancer, there are 4 different subtypes of breast cancer, and if you treat someone with the wrong medicine that has the wrong subtype, then you are essentially not treating their disease. And many other types of cancers have a lot of very particular subtypes. So a gene has a mutation, and that mutation causes something to break, and if you treat it for the wrong gene that's been mutated, you're not treating the right thing. So personalized medicine in this context is using data about an individual or data about an individual's specific subtype of disease in order to treat them better. So instead of a doctor just looking at them and using their experience in the context of genomic personalized medicine, the doctor will be able to use a biomarker test, whether it would be an assay in a laboratory or a SNP chip, or potentially at some stage using the sequence of their genes or the sequence of their cells. There are a number of IP issues related to personalized medicine. Electronic medical records are being increasingly used for personalized medicine, so that the bulk of the data that doctors can collect from patients can be used to better develop a body of knowledge related to different diseases and types of diseases. So instead of having all of the patient medical records on paper in a doctor's office, they can be accessed potentially by an entire hospital system and so forth, or enable a new drug to find the right patient cohort for a particular study. I'm going to touch upon an idea called Pink Army, which is a biotech biopharma co-op. So instead of having a for-profit company that is in the business of disease management, so if they actually develop a cure for a particular disease, then they lose a customer. In the co-op setting, an individual can buy into a biopharmaceutical company and actually have joint ownership for a product that's made to treat their disease based on their own cells and their own genotype. There's a lot of interest in nonprofit clinical research organizations, and the big data aspect of this is the electronic medical records, personal genomics, and other types of bioinformatic data. So this talk is, I feel, a little bit out of place at the OSCon Data, given the talks about Facebook and LinkedIn and HBase and Hadoop and PyPig and NoSQL and so on. But what would a social web app be without the people on that social graph. What is life? What is artificial life? And incidentally, what is consciousness? These are some interesting definitions of ways that one can quantify consciousness. There are many different ways to sort of typify what one would mean by consciousness. One scholar says that a brain is a state machine. It contains an inner neuron partitioning conscious and unconscious states, perceptual learning and memory, prediction, self-awareness, representation and meaning, learning utterances, learning language, will, instinct, and emotion, and another has another set of concepts. So based on these ideas of what consciousness is, clearly a lot of synthetic biology is nowhere near conscious. It is, however, artificial life. So artificial life is, or A-life as it's sometimes called, is an area where computers mimic the processes of life. They mate or they exchange their genetic information or whatever. Or, an existing organism is changed in such a way that it has been designed. And in a sense, it's artificial because it's been changed by an outside entity which has control over them. In addition, I think that this topic has particular relevance to open source because, in a sense, life is open source. You can read the sequence for life, you can read the sequence for proteins, you can read the sequences for our genes. And we should have excellent tools to manage our source code, so to say. So as this area has been maturing over a couple of years, a number of web tools and sort of consortia for managing new things have started. One is Open Wetware, which is a wiki whose goal is to promote the sharing of information, know-how, and wisdom amongst researchers and groups who are working in biology and bioengineering. And from the very beginning, this whole area and the people working on it were really deep in open source. Many of them were computer scientists who used Linux or open source tools. And the tools they use for their software is also open source. If anybody has any questions or wants to make any comments, feel free to speak up. Or clarification, whatever. The BioBricks Foundation is related to Open Wetware, and it relates to these individual components that are subsequently assembled into larger synthetic organisms. It provides a bunch of, it runs in part the iGEM competition, which is this yearly competition where tens of teams from universities around the world come and compete to engineer E. coli that smell like bananas, or organisms that secrete a siRNA that is relevant for breast cancer, or any of a variety of things. I'll talk about a couple iGEM projects. There are too many to go into great detail on, but I picked two that I think are interesting too. So this is iGEM. iGEM is the International Genetically Engineered Machine Competition, and it's the premier undergraduate synthetic biology competition. It's a pretty cool thing. It's been going on since 2004, and it just gets bigger and bigger every year. Originally it was a handful of teams, like 4 teams, and now it's, I think, 1,000 people, and they're trying to break out and have a iGEM competition in Asia and an iGEM competition in Europe, and probably they'll still have the iGEM competition in Boston for the grand final. One of the most interesting things that's been coming out recently is so-called next-gen sequencing. So next-gen sequencing is using either solid-state or non-solid-state PCR type tools to get sequences for people or for bacteria or for viruses. And a number of different companies are in this field. Illumina, Applied Biosystems, Helicos, Roche 454 are a couple of them. And they all have different characteristics. The throughput statistic varies widely. The data per run in gigabase pairs varies widely. They're just different companies have developed different engineering paradigms for sequencing stuff. For a long time, sequencing was much too expensive, and SNP chips, or single nucleotide polymorphism chips, have been used extensively, while next-gen sequencing was just becoming more popular. So, for instance, a lot of the tumor data that I work with is all SNP chip data. So, basically, what you get is you get 500,000 probes of genetic markers on an individual, and you can get a pretty accurate genotype of that individual or a copy number profile of that individual using these chips. So instead of it being $1 million to sequence someone for $450 or for $250, you can actually get a pretty high profile or resolution of that individual. So 23andMe, which you may have heard of, is using SNP chips. It's using a different type of SNP chip. It's using an Illumina SNP chip that, in the third version of 23andMe, has, I think, 1.2 million probes, and the per sample price is $250 to Illumina. Yeah, so these are some of the things that you can glean from a SNP chip. You can get a copy number profile, which is the number of copies of different genes. You can get gene expression on gene expression arrays, so you can look at expression levels of particular genes. In some cases, overexpressed genes can be indicators of particular things. You can get a major copy proportion. Many people do machine learning on SNPs because basically it's just an array. So you could have 10 patients and you have an array of 250 SNPs, and it's just a vector. So you can do machine learning on these vectors. You can do support vector machines, Bayesian networks, Markov chains. Biomarker identification is the process of finding a set of characteristics that characterize a particular disease. And for instance, someone who would be susceptible to a particular therapy, one would look for a biomarker for that sensitivity. There was a recent study, but it's a very broad term. Recently, a study came out that said that a biomarker for autism is simply a lack of empathy. There is no genetic biomarker against lack of empathy. It's debatable, obviously. And there's a wide spectrum of autism as well. So where are we? How much do we know about cellular processes, regulation, oncogenes, protein-protein interactions, microbiome interactions? How much do you know about brain structure-function relationships, gene activation in neurons, memory, electrophysiology, neuronal cognition, or brain maps from worms to mice to humans? I would argue we know a lot, but still nowhere near the entire set of information we would like to know in order to be able to design therapies or simply understand these different things. So if you are curious to download the human genome, it's actually quite easy. Just go to ftp.ncbi.nih.gov, and you can download this complete sequence for a particular sequence of the human genome, and it's right there. So as synthetic biology has been maturing from 2004 and 2003 to this paper was published in 2009, a lot of emphasis has gone from just building these little blocks like a transistor or a OR gate or something into building systems much like a circuit board, and figuring out ways to isolate these different systems from each other using insulators or other things, or just characterizing the functional characteristics of the gene interactions or regulatory system interactions. So this is a figure that's talking about modules based on transcriptional, translational, and post-translational control, and in particular feedback loops in these systems. So The top one is an RNA device that contains a ligand, the catalytic region, an aptamer, and a sensor. And this other one has a leucine zipper and another system. And so one of the things that's happening is people are starting to build like data sheets. So if you ever did any electrical engineering, what you do when you get a little integrated circuit, and you want to figure out what the pins are, you get the data sheet for that thing, and it will tell you that pins 5 through 6 are 5 volts or whatever. And so building data sheets for these modularized parts is growing rapidly. This one has a public license, and it's from the Registry of Standard Biological Parts. And they tell you how to make it, and what the static performance is, what the dynamic performance is, what the input compatibility is, what the reliability is, and the operating conditions.
Question?
Yep.
So I get the impression that the idea here is to, at a biological level, reinvent the usual modules of electrical engineering. You know, gates and flip-flops and things like that. You might build a computer. Is that true?
To a certain extent, yes, that's true. It turns out to be very difficult to do a lot of chained AND gates, for instance. Like having 3 or 4 chained AND gates is about the state of the art. So basically, you end up having to do different things because the environment is so different.
Well, I know that Carver Mead, integrated circuit guru, is actually kind of critical of digital logic in that you spend a lot of time and energy overcoming the natural capabilities of silicon, and argues that we should design systems in an analog mode that exploit all the properties of gates instead of overpowering them to make them digital. And I wonder if there is a design style emerging that is more natural in that sense, in a place where digital might be even harder.
Yes, definitely. There's definitely a design style that is a biological design style that is more akin to analog electronics, with curves like these. And instead of, you can't get a spike, you can't get a.
Sorry about that.
Yeah, I mean, you don't have a clock for one thing, so you have to. It's just different. So partsregistry.org is a place that one can find these things. So along with the individual parts, a lot of tools are emerging in sort of the systems biology world that are based on things like the systems biology markup language, and in particular, there's a tool called Cell Designer, which allows one to design using a graphical environment what you would like to do in the wet lab, using abstractions and simulating things a little bit to see what the right concentrations should be for the output you desire, just to sort of allow one to go one step further than what you can design in your head, to be able to do some experiments on a computer system. I'm sorry?
This is open source software.
Yes, this one is. So in the system biology markup language, it interfaces with a number of databases, from the PubMed database to KEGG database, and then you have software tools, and you can design cells in this way and plot behaviors you've simulated, change the simulation paradigm, and so forth. So in addition to these types of synthetic biology things, there's also a lot of work going on in structure of DNA molecules. So in this case, there's a nanorobot that's been made out of DNA molecules that can encapsulate a payload, and in this case, it's been designed to treat leukemia cells. So it recognizes a cell surface receptor, it has a gated function, and then it releases its payload. It has essentially a one-step logic. It recognizes something and does something. Unlike in the case of many small molecules, they're bound to a ligand pocket and inhibit a protein or something like that. So 23andMe and next-gen sequencing and SynBio getting together can do some really cool things. So this is the 23andMe version 3. It uses the Illumina Human Omni Express feature, which is $250 per sample. And it has 700,000 SNPs, and you need 200 nanograms per sample. So just like Cell Designer, a lot of new computer automated design and biological CAD tools are being developed. AutoCAD in particular has a new group in matter design, and in some, they're trying to work a little bit more in synthetic biology topics, the AutoCAD company. So Thingiverse is a place where you can download open CAD models. There are increasingly many 3D printers, laser cutters. You can send designs off to be printed very cheaply and of high quality. There's more personal fab technology. The MakerBot, which is a cheap 3D printers, and other types are becoming more widespread. I think this picture is actually from OSCon 2008 or something. So this is the RepRat matter compiler, which is a particular matter, so-called matter compiler. It's just an extruding printer, which can print all of its own parts except for a couple that are like metal dowels that you can buy at any hardware store. So the idea is that with this printer, it's actually self-replicating. So you can so it behaves like bacteria sitting on food. It will just be able to replicate. It can print itself, basically. So there's no constraint of having to have a factory to build these things. It can print itself. Now we have a movie. So this movie is a description or an animation of apoptosis, and I just wanted to show it to you because it elucidates some of the systems that are going on. Apoptosis is the death of cells. So there's a disease cell, these are healthy cells, and there's a killer T cell. So these are the cell surface receptors that the DNA nanobot that I was talking about latches onto.
Now, is this from an actual simulation, or is this just a representation?
It's not a molecular dynamics simulation. It is a simulation. Wasn't that nice? Okay, so like the gentleman asked, the way that these are simulated typically in a biophysics setting is you do what's called molecular dynamics. So you integrate the equations of motion, you have a force field that says how protein should interact with other protein molecules, how water should interact with the solute, and so on. In addition, you can do mixed quantum mechanics, molecular mechanics simulation. So you have a little box that's a quantum mechanical simulation, and it's joined to a molecular mechanics box via kind of a switching function. And in addition, there are a number of biological pathways, chemical reaction simulators, and optimizers, and uncountably many projects in gene network analysis and stochastic simulation. So in case it isn't clear, I think that the marriage of data and first principles and functional abstractions can do some really awesome things. So if you're able to build a sensor that can sense what's wrong with the system, and you have enough data about the system to know that that is a unique characterization of that thing that's wrong, then you can do something about it. And so in this case, if you have SNP information, or you have gene expression information, or you have from the next-gen sequencing, and the marriage of electronic medical records and a patient's sequence, you can potentially design whatever therapy you want, really. So instead of having to spend 10 years and $1 billion to get a small molecule through FDA approval, I think that there are options. Very few small molecule drugs have been approved in the last 10 years or so. So how and where are these data used or made more usable? Randomized clinical trials. You can do biomarker development, genomic tests in these certified labs. The licensing, these developed biomarkers can then be licensed to the companies for marketing and development. So in the practice of doing these kinds of biomarker development, a lot of databases are used. There's a database called GEO, the Gene Expression Omnibus, which is a NIH-funded database of genomic information from papers that have been funded by the government, which allows reanalysis of countless data sets to be done. So ours is a common package that's used. Bioconductor provides a lot of tools to allow biological analysis of these gene expression sets or SNP sets or sequences, and a variety of statistical methods. So that FDA approval for a process, not a drug or biological device, is a way to make this kind of personalized medicine more scalable. So there are a number of cellular therapies, particularly for melanoma and some other cancer therapies, where basically there's a process by which cells are taken out of the body, saved, chemotherapy is given to the patient, and then the healthy cells are reintroduced, among a number of other ways this is done. And so this is a process, it's not a drug, a biologic, or a specific device. So back to Pink Army. So in the Pink Army cooperative scenario, you own your own cells. Why can't you control what's done based on your own cells? So you can develop therapies based off of your own cells, and they should be yours in the co-op, not owned by a for-profit company. And if a biopharma company cures your disease, they lose a customer who's cured. So it's relatively inexpensive to do a lot of laboratory work, and if the procedure is worked out satisfactorily and is proven to be safe and efficacious, and the other constraints for phase 1, 2, and 3, and 4 trials by the FDA, then a model like this one could potentially occur. This is some data on cancer 5-year survival rates. Pancreas and lung cancer have very, very low survival rates. Prostate and melanoma are much higher. So just as some data points on how much it costs to do certain CRO trials or clinical research organization trials. For a melanoma trial, with only 1,000 patients, it would cost about $60 million in CRO expenses. For a chronic obstructive pulmonary disease drug, it would cost between 240 and 250 million for 10,000 patients in just CRO expenses. So oncolytic viruses are viruses that would cause cancer cells to lyse. It's very difficult to get lysing viruses approved for much of anything. But given the fact that you can make logical things, basically if it sees this, it can do that, or it can do that, or if it doesn't find it, it can kill itself. Basically, they're not computational devices. You're not trying to compile something, but it can perform logical functions very slowly. So, for instance, in one model that we're working on right now, you can use data to engineer therapies or cures for genetic diseases. So if you have a nanoparticle and it sees a particular combination of RNAs at a particular time, then it can do something, like release a protein initiating apoptosis in the tumor cell. And these logical ANDs can be ORs, or you can have many more of them, or many fewer. It's not limited to 5. You could have 2. You could have 8. Do you want to see a movie of tetracular robots?
Hey, I got a question. What about non-proteins that are like transfer RNA or anything else? Do you control those as well too?
I'm sorry, I don't understand the question. What do you mean by control?
Well, in the previous slide, you talked about RNA. What about like non-proteins or transfer RNA or small nucleotide RNA? What about those? Are those part of it?
In the particular sensor that I was referencing there, it's specifically a short RNA sensor that's not sensitive to single nucleotide mutations. So it can do a matching between short RNAs, but if there's a single mutation, then it actually can detect a single or sometimes a double mutation. So there's some more biological nanobot technologies. This is DNA origami. So these are actual DNA molecules that have been, where their sequence has been engineered such that they can make smiley faces or pyramids or stars. Obviously, the biofuel community and ExxonMobil have been very interested in this for a while. There was a $600 million contract signed between Exxon and Craig Bender's company, Synthetic Genomics, to develop a synthetic biofuel technology. And these are just a couple of the iGEM things. So they realized that you can make E. coli glow, and they were like, well, why don't we make a light bulb out of E. coli? And they call it E-glowing. It's a bioluminescent picture. And some students at the Chinese University of Hong Kong were very interested in a bio-hard disk. So you encode everything, well, bits as AT, C, and G to represent numbers. And in this model, insertion in a cell of the United States Declaration of Independence is 18 cells. And so they they design these parts and they put them back into the repository of parts as part of their project. And on the biohard disk, 1 gram of E. coli is equal to 900 terabytes. Very big biological data. There's also a lot of interest in microbiome engineering. So basically the microbiome, or the set of microbes inside your body, could potentially be engineered so that they could actually provide you with a continuous source of drugs or elements that would cure a number of genetic diseases. So for instance, engineering a microbe that's introduced into the microbiome that produces interleukin-10 would essentially cure many of the symptoms of Crohn's disease. You could engineer a microbe that makes insulin for diabetics. It's a highly competitive environment, and it's an active area of research to do this. In addition, in the chemicals industry, people are interested in cleaning, refinement, scalability, and more sensors. Conclusions. I hope I have explained some wide-ranging implications to existing problems in health, engineering, energy manufacture, particularly human healthcare, genomic research, and data-aided design and testing, and also some new paradigms for manufacture processes and biological design concepts. I'm happy to take any questions. Yep.
It seems like a lot of the tools that you talked about and a lot of the things you were describing were techniques or tools for looking for basically positive effects in these things you construct. The question is, what kind of tools are available for negative effects testing? So like what kind of testing is done for side effects, undesirable effects, before these things are actually used, before these things are actually, implemented in the lab or something like that?
In a lot of cases, the side effects can be tested using existing laboratory equipment. We don't need to design something new to do that. But certainly you could have a micro. I mean, there are many of the IGEN competitors have made sensors that detect toxic levels of different things, and so just having an engineered microbe that detects a toxic level of something that was generated by something that was synthetic is very practical.
But no, like software tools that do that or anything like that.
You would just set the parameters different.
What was that?
You just set the parameters different. It's the same tools. You set the parameters different to get your numbers.
Sure, but I mean, when you're talking about testing for, say, 500,000 negatives and 2 positives, we need sort of different software to do that. You're not going to cover 500,000 negatives, right?
I would use the same software.
Is that what you would do?
It really depends on the situation.
Yeah, I guess my question was more, do you know of any tools that exist that do something like that? Or is that just, like she said, like you would just sort of code up nukes and just to test them?
No, I mean, it's very simple to make a sensor. I mean, if it sees something, it does something. And that's the same paradigm whether it's polymer or not.
So nothing specific for that?
I don't think it's a specific scenario.
Okay.
I think I'm fine, so I'm happy to take any questions offline as well.